that skydiving with you too.
Yeah, that was a lot of fun.
Oh, parasailing. I guess it was parasailing.
Wait, we have to hear a little more about that, Rick. What was that? You guys were skydiving?
After DDW, Carol and Dennis and Finley and Marley McCrae stayed with us for a couple of days, and we went out to the glider port, and there's a cliff, a 400-foot high cliff over the Pacific Ocean, and these guys come out, and you can rent a teacher to sail with you, and you jump off the cliff.
Oh my God!
Yeah, and then you sail around for a while. Yeah. Oh, it was fantastic. So Dennis was on one side, I was on the other side, and we like for a half an hour we flew the oceans.
Unbelievable.
Yeah.
It was great. These boys are always up for an adventure, that's for sure.
Boys will be boys.
I know. Some boys never grew up. Just like Peter Pan.
Right.
All right.
I thought that was a good memory.
Yeah, you bet.
It's just a little after 7, and I think it would be nice if we started the second annual Dennis Ahnen Lectureship on time. So it's going to give me great pleasure to welcome all of you to this special event. I will tell you that I had a very personal conversation with Dennis earlier this year, and he actually helped me come up with a list of who would be giving the lectureships. And he specifically wanted Rick Boland to give this lectureship this year. So we're very, very, very pleased that Rick's been able to join us. I think as a division, we miss the continuous optimism, enthusiasm, and scientific process that Dennis has brought to us every Friday. And we will survive, and in his memory, we will try to do our best to continue a very effective Grand Round series. So without any further delay, I'd like to introduce Swati Patel, Dennis's protege and honorary family member of the Ahnens, to start off the lecture this morning. Swati.
Thank you, Steve. Good morning, everyone. I'd like to welcome everyone as well, especially Dennis's family who's here joining us, his wife Carol, his daughters Gina, Megan, and Nicole. We're so glad that you're able to join us and hope that you'll join us every year as we honor Dennis. I'll be introducing our guest today, but before I do that, I just have to take a few moments to talk about Dennis. As many know, he passed away earlier this fall after a year-long battle with appendiceal cancer, an unfortunate, cruel hand to be dealt to a man who really dedicated his life to cancer prevention. I think Dr. Ahnen honestly would be kind of annoyed if I reiterated his curriculum vitae and his myriad accomplishments, contributions to our field. So instead, I'd like to share this word cloud. He really loved word clouds. He thought that they really just visually represented so many ideas. And I've presented this word cloud before, and it was actually generated when he received a Lifetime Achievement Award for our Hereditary GI Cancer Society, the collaborative group of the Americas. And it was really from words that many of his friends, colleagues, mentors, mentees, collaborators from around the world, the top words that came up when they thought about Dennis. And I think it just really encapsulates this man. I've been on the inpatient service this week. As you can tell, I'm in my greens right now. We've had some exceptional patient care with our first year GI fellows being very, very busy. And as I was thinking about this lectureship this Friday, I was a little bit saddened to think about the rising generations of trainees, junior faculty who just won't get to experience Dennis's sparkle the way many of us tuned in today have been honored to do so. And really, it's not just those of us tuned in today, many faculty here, fellows joining, but people really all over the world that have been able to benefit and really rise because of Dennis's mentorship and collaboration. But honestly, of all the impressive contributions to science, the accolades, the awards, he really told me that one of his most proud accomplishments was establishing this lectureship. He told me that he really couldn't think of a better way to honor his legacy than to provide a mechanism to invite world-renowned speakers, world-renowned experts to really honor his memory. And I think fellows and rising generations will always get to experience Dennis's passion, his generosity, his kindness, and his sparkle through this lectureship. And so he had the forethought that maybe none of us really appreciated at the time in really advocating for this lectureship. And for that, we will always be grateful. It really gives me great honor to announce this year's visiting lecturer, Dr. Clement Richard Boland. He's a professor of medicine at the University of California, San Diego, has a storied career and continues to have a storied career. It's really hard to know where to begin to introduce a man whose CV is 58 pages, single-spaced, over 400 peer-reviewed publications. Proud to say I've co-authored 5 of those with him. He's received the highest honors in our field, including the AGA William Beaumont Prize, the AGA Friedenwald Medal, as well as the Collaborative Group of the Americas Lifetime Achievement Award. He's mentored hundreds, over 100 physicians and scientists and trainees, many of whom credit Dr. Boland with providing the pivotal foundation they needed to build successful careers. He's been a leader in our community at both the institutional level with division chief, certainly on his track record, but also within the larger GI community and service on the national level. He's been really a tireless advocate for research and for quality patient care through the AGA, has served on multiple committees, the editorial board of gastroenterology, and also, of course, serving as the AGA president in 2011, 2012. Dr. Boland has made groundbreaking contributions to the field of familial and hereditary cancer. And his research interests really have focused over time in DNA mismatch repair in colorectal cancer, tumor genetics, and the mechanisms of genomic instability and tumor progression. But honestly, perhaps what's most unique about Dr. Boland is how he got interested in this field. And we were talking a little bit about how Dennis got interested in the field early on. And it's really, I think, wonderful to hear about his personal connection to his life's work. So I'm looking forward to hearing some of Dr. Boland's personal reflections about the field of hereditary cancer and how it's changed since he first began his career. So on that note, Dr. Boland, we want you to know, of course, just how grateful we are that you're able to join us today in Dennis's honor. As Steve mentioned, pandemic or no pandemic, it was Dennis's specific request that you share your wisdom with us this year in his name. And, although we're certainly saddened that we can't host you in person and show you Denver, spend the day with you and give you a tour of our institution and have you meet all of our faculty and fellows, we still are so grateful that we have this platform to share this experience. So I will go ahead and start sharing your sides, Dr. Boland. We're really excited to hear about your talk today on Lynch syndrome and beyond. For the audience, please feel free to enter any comments or questions in the chat box. At the conclusion of his lecture, we'll have a question and answer discussion session. And then Dr. Boland has very generously agreed to spend some time with our GI fellows after his grand round session this morning so that they can hear about his life, his career, and his approach to mentorship. So please join me in welcoming Dr. Boland here.
Thank you. I'm going to try to share my screen now. Is that working?
We are seeing your desktop, yes.
Okay. Is that working too?
Yep, that's perfect.
Great. Can you see the cursor?
Yes, we can.
Great. Okay, so let's go. First of all, this is really dedicated to Dennis. That's what this is all about. And I want to give a big shout out to Carol, Gina, Megan, Nicole. You guys are wonderful. Dennis was such a good friend. And a day doesn't go by that I don't think of him too. Dennis and I met at DDW in Salt Lake City in 1981. I was in line at McDonald's with my mentor, and we were talking about cancer research, and I'm not making this up. And Dennis turned around and said, "Hey, I do cancer research too. We need to know each other." And the first paper that I wrote without my mentor was with Dennis. We actually did a project together. I'll always miss the guy. We can't really replace him. But in any event, I had my own reasons for getting into cancer research, much as the way Dennis did too, at least with time. And it had to do with the fact that I came from a Lynch syndrome family. Here in my disclosure is my one disclosure. So let me talk, what we're going to talk about today is how do you evaluate a familial gastrointestinal cancer syndrome? So you suspect this kind of thing when there's a positive family history, when a person has an early age of onset of cancer, when there are multiple primary tumors in one person, lots of polyps or unusual pathologies, or when you screen a cancer and it has microsatellite instability or abnormal immunohistochemistry for mismatch repair proteins. So how do you size it up when any of those things pop up? Well, the first thing you do is you have to decide, are you dealing with polyposis or non-polyposis? But there is some overlap. So some polyposis syndromes don't have that many polyps, and all of the non-polyposis syndromes really start with polyps. So what we have here is, on the one hand, we used to think that the only polyposis syndrome was FAP. And we also knew that there were these rare hamartomatous syndromes that had unusual, unique histologies. But now we know that there are recessives. And we used to think that about a quarter or a third of FAP was a new mutation, but it turns out that some of them are actually recessive. The interesting thing about recessive diseases, they're completely genetic, but they don't look very familial. And so these genes can cause those. And then there are these more recently discovered oligopolyposis syndromes like constitutional mismatch repair and polymerase proofreading mutations. More common by far than the polyposis syndromes or the non-polyposis syndromes. Mainly it's Lynch syndrome, Lynch-like syndrome, and familial colorectal cancer type X, and we'll go into all those. So Lynch syndrome is the autosomal dominant familial disease caused by pathological variants in a DNA mismatch repair gene. And the 4 genes that are involved are MSH2, MLH1, MSH6, MS2. Also EpCAM, which is the immediate upstream gene in front of MSH2. When you lose the stop codon, it will silence MSH2. So that acts like MSH2 Lynch syndrome. And the tumors, by virtue of the absence of a mismatch repair gene, they have microsatellite, somebody's not silenced, by the way, microsatellite instability and deletion of the expression of the responsible mismatch repair gene. And this predisposes to colorectal cancer, endometrial cancer, ovarian cancer, and some other cancers too. And the tumors are all hypermutated and immunogenic. So we'll get into this. So the first thing we want to think about is the mismatch repair system. So the mismatch repair system, it's a DNA repair system that hooks on to the DNA polymerase. Well, no, it doesn't. It hooks on to, it senses when the DNA polymerase has made an error during synthesis. So there are multiple enzymes involved. There's the mismatch repair recognition proteins, which are the MUT-S heterodimers. There's the recruitment proteins, the MUT-L heterodimers. And then there's, when these recognize the error and recruit the repair system, then you get long patch excision. So you excise everything that was been made there and then resynthesize the DNA. So this is what the mismatch repair system looks like. So let's say you have a mismatch. You put a T, the DNA polymerase makes a mistake, puts a T across from a G instead of a C. So then the MUT-S heterodimers will recognize that. And that's made up of MSH2, which is right here, which will heterodimerize with MSH6 and that's called MUT-S alpha. There's also another pair that can occur, which is MSH2 and MSH3, which has a slightly different recognition specificity. And when they bind to this will recognize slightly longer mispairings. And then they recruit the MLH1 and its partners, either MSH2 or MLH3, and that recruits a whole bunch of other proteins that will excise what the DNA polymerase has just made. And then it will pop off and the DNA will be resynthesized. But the key is that these are all functioning as heterodimers. MSH2 can partner with either of these two. MLH1 can partner with either of these two. And if they're not dimerized, the proteins are unstable. So what that means is that if you've got a germline PV's pathological variant in MSH2, then the immunohistochemistry will be negative for MSH2 and MSH6, but you'll have the other two proteins will be there. So now you know if you see these two are missing, you know that MSH2 is the problem. In the case of MLH1, if you're missing that, if you've got mutations in that, you will not see MLH1 and PMS2, but the other two will be there. And for these two minor proteins, if they're mutated, then you just get one abnormal immunohistochemistry. So that's where that's how you interpret the immunohistochemistry. So how prevalent is Lynch syndrome? Well, about year 2000, I said, well, if 5% of the population gets colorectal cancer and 3% are Lynch syndrome, if the penetrance was 50%, it would be 1 in 333, or 1 in 350, somewhere like that. In fact, I used to make the joke that there were 351 people in my high school class, and I used to say, well, so somebody in my high school class had to have this disease. Well, okay, so, but then as the information came along, in Iceland, they have DNA on everybody in the population, and they use something called imputational genetics, where they use the haplotypes, and they recognize that 1 in 226 have Lynch syndrome. Now, it turns out that not that many people get colon cancer because there are important founder mutations in these two genes, and as we're going to see, these are relatively less penetrant. Well, in other words, the people who have the disease don't get cancer if it's not penetrant. And now we know that in Australia, US, and Canada, it's 1 in 279. So I was actually too modest in guessing 135 or 1 in 350. It's 1 in 279. And it turns out that the real reason is because a very large proportion of Lynch syndrome is due to mutations in MSH6 and MSH and PMS2, which have a lower penetrance. Now, there's a lot of clinical heterogeneity in Lynch syndrome. First of all, you got multiple genes that give rise to the phenotype. 5 genes produce one disease, and the penetrance is incomplete, and it varies by gene, as we're going to see in just a minute. So some of the Lynch syndrome genes are more highly penetrant, some less so. MSH2 gives the full spectrum of tumors. About 50% will get colon cancer, about 70% will get any kind of cancer. And then EpCAM, the upstream gene, will lead to a similar type of disease. And then MLH1 gives the full spectrum of tumors, 50% colon cancer, 70% any kind of cancer. And then MSH6 has a lower penetrance and later onset of cancer. So, you know, a person can be 60 or 70 years old and get a colon cancer and can still have Lynch syndrome if it's this disease. It has a very high penetrance for endometrial cancer, though. And PMS2 has a much lower penetrance for cancer. And as we're going to see, it's kind of almost a different disease and a lot fewer in terms of gynecological cancers. So do we manage all our patients with a positive family history the same way? Of course not. We have to take a dive and find out what's really going on. This is an interesting study. It came from Finland in year 2000, and it was a 15-year prospective study. It was it was a controlled study, but non-randomized. So in Finland, Lynch syndrome is particularly caused by a couple of big founder mutations among Finns, because not that many people move into Finland, not that many Finns move out. And so they made a controlled study by the controls for people who refused to have colonoscopy or didn't have access to it because they live, up in the tundra. And so what happened was that they were screening them with every 3-year colonoscopies. And when they screened them, the proportion of people who were free of colon cancer was pretty high because they were taking out all these polyps. And this is the control group. So the people who were screened had a 94% likelihood of getting a cancer over 15 years, and the controls, it was 83%. Then genetic testing came along, and the people who were mutation positive versus mutation negative had a huge difference in terms of being cancer free. So so it really made a difference once they were able to find who carried the mutation in the genes. And we're going to dig into this in a little bit more. And this is where the lesions were. These were the mutation positive patients had a lot more lesions than the mutation negative. So there's a lot of familial colon cancers we're going to see in a minute that is not Lynch syndrome. And they had fewer cancers, and they were mostly all adenomas. So here's the cumulative incidence of cancer in Lynch syndrome by gene. So MSH2 and MLH1 are pretty similar. So the likelihood of getting, this is from a prospective study from the Mallorca group that was just published 5 years ago. And the colon cancer incidence between ages 25 and 70 was here for MSH2 and MLH1. Endometrial cancer is a big problem in this disease too. This is among women, of course. Ovarian cancer too, and any cancer, these two diseases, it was 72%. But for MSH6, there was a lot less colon cancer. There was a lot of endometrial cancer and about a 54% likelihood of any kind of a cancer. And for PMS2, it was way less, and no ovarian cancer. So our guidelines as we go forward are going to be based upon not just whether it's Lynch syndrome or one of these other mimickers that I'll tell you about in a minute, but also what genes involved, because you're going to manage this kind of problem differently than you'll manage this kind of problem. And in fact, just last week, new guidelines came out from Europe that said that they would start screening Lynch syndrome at age 25 if it was MSH2 or MLH1, but 35 for these two genes. And you might even spread out the intervals to maybe 2 or 3 years for colonoscopy. So let's take a look at this is a study from Europe that came from Germany, Netherlands, and Finland. And this is the cumulative incidence of adenomas by genes. And as you can see, whether it's MSH2 or MLH1 or MSH6, they all get a certain number of polyps as time goes by. Interestingly, for advanced adenomas, MSH2 is a little bit worse. And we're going to take a look at why that might be in just a minute. And for cancer, MSH2 and MLH1 have virtually the same incidence of cancer. MSH6 is less, and PMS2 was not studied in this, but it's even less than that. So we're looking at different degrees of risk based upon what gene caused your Lynch syndrome. So just to summarize colonoscopic surveillance, it prevents some but not all colorectal cancers. It prevents fewer cancers in Lynch syndrome compared with non-Lynch syndrome familial colon cancer. And I took a slide out in the interest of time, but there was a study in Holland in which they had a group of people who were being studied, who were being surveilled every 2 or 3 years with colonoscopy. And when the testing became available for Lynch syndrome, it turns out that by doing colonoscopies in people who didn't have Lynch syndrome, you brought the incidence of cancer down to 0. But the Lynch syndrome patients continued to get cancers, but they didn't die of it. So it's actually colonoscopic surveillance is way more effective when it's not Lynch syndrome, because the Lynch syndrome tumors, they develop too quickly. And it turns out that every 3-year colonoscopy reduces colorectal cancer by 62%, and it's less effective than every 1 to 2 years. There's a debate about whether to do a colonoscopy every 1 or 2 or 3 years. Probably every 2 years is effective. But the patients will continue to get cancers, they just won't die of it. And we're going to talk about how these tumors develop and why there might be a difference in cancer incidence based upon how you screen. So to do that, we have to think about how do tumors form in Lynch syndrome. So first of all, a person has Lynch syndrome if they have a germline pathological variant in a mismatched repair gene. So you don't have to have cancer to have Lynch syndrome, you just have to have that mutation. The lesions, the adenomas and carcinomas are more frequent, appear earlier. The adenoma to carcinoma sequence accounts for most of the colon cancers. Interestingly, sporadic cancers usually develop through mutations in the APC gene or the beta-catenin gene, but turns out that the RNF43, it's a ubiquitin ligase that regulates the Wnt signaling. It's got some mononucleotide repeats in it, and in Lynch syndrome, this thing gets mutated a lot. So that's probably a more common way for cancers to develop in Lynch syndrome. Also, the fact that people get more polyps earlier in Lynch syndrome, even before they have the second hit that occurs, suggests that haploinsufficiency. In other words, when you have Lynch syndrome, one gene is mutated, one is wild type, but that apparently produces less of the mismatch repair gene, and that may be permissive of somatic mutations, and that might be why the adenomas occur earlier. It's the second event, the somatic event in the polyp, where the mismatch repair gene leads to mismatch repair deficiency. And what happens is that adenomatous polyps are, when they're small, they typically don't have mismatch repair deficiency, but once they get to be about 8 millimeters in size, then they're mismatch repair deficient and they've gotten that second hit. That's what that's all about. And this leads to a hypermutated phenotype. So they accumulate mutations at a very high rate. And we're going to talk about mismatch repair deficient crypts, which would permit a de novo non-polyp mechanism for some cancers to develop in Lynch syndrome. So if we take a look at the second mutational event, so the classic Knudsen hypothesis was, and he came up with this idea by studying retinoblastoma. You get a germline mutation in the retinoblastoma gene, and then you need a second hit. You're not born with a tumor in your retina. You need a second hit. So the wild type gene has to get a mutation, and then you get the tumor. But people who have hereditary retinoblastoma have an earlier onset and are more likely to get tumors in both eyes. So that turns out to be largely what happens in most tumors. And so we have here the adenomas, you see a germline mutation and loss of heterozygosity. That's the second hit, or a mutation. And that occurs fairly typically, not every single time. There's also other things that can happen, which is like methylation of the gene, or that can happen too. Now, in the case of Lynch syndrome, there's this idea that was discovered by Matthias Klur from Heidelberg. These are mismatch repair deficient crypts. Let's take a look at this. They did systematic analysis of non-neoplastic colon and small intestine in Lynch syndrome patients from the resected specimens. They did look for microsatellite instability. They did immunohistochemistry, and they analyzed target genes for mutations, looking for the earliest abnormalities in Lynch syndrome. So here's a stretch of colonic epithelium, And here's the H&E. And on this side, this is a mismatch repair deficient crypt. And on this side, this crypt looks perfectly normal. There's some minor abnormalities here, but nothing that's neoplastic. So they did immunohistochemistry for MLH1, which was the gene that caused this person's disease. And what you see is that the positive staining is when there's dark staining in the nuclei. So there's a little bit of dark staining here, a little bit of, that's normal. And the middle of this crypt expresses no MLH1. So that's a typical Lynch syndrome finding, failure to express this particular mismatch repair gene. And they look, this is a proliferative marker, and there's no hyperproliferation. So there's a little bit of distortion here, nothing neoplastic, no expression of the mismatch repair gene, and no hyperproliferation. And I'll go over this quickly. Essentially what this shows is these are markers for microsatellite instability, but these mismatch repair deficient crypts have microsatellite instability. That's what this shows. And also it's got mutations at the TGF beta receptor 2. In other words, it's got target gene mutations. So it's got all the things you would expect if you wanted to have a neoplastic lesion, but they're not neoplastic. So in defective mismatch repair crypts, you have isolated crypts that lose mismatch repair activity. It's 1 in 10,000 crypts lose mismatch repair protein expression. They have microsatellite instability. There are mutations at the cancer-causing genes, but they don't look dysplastic or neoplastic, and they're all over the place. There were 8 out of 10 Lynch syndrome colons had this, 1 crypt per square centimeter of colon, 1 for every 2 square centimeters of small intestine. Now, what's interesting is people with Lynch syndrome don't get hundreds or thousands of cancers. They get 1 or 2 or 3 in their life. So these defective crypts are far more common than neoplasms. So I think that most of them are shed. But what's particularly important is that these crypts generate immunogenic peptides. So there's microsatellite instability, and that instability particularly occurs at simple repetitive sequences called microsatellites. And you get a frame shift, which is loss of typically 1 of the nucleotides. And then you get the frame shift causes all the downstream sequences shift 1, and you get the same immunogenic protein over and over again through your whole life. So through the entire life of a person with Lynch syndrome, they're immunizing themselves against these frame shift peptides. So that's probably why the outcome, as we'll see, is better in these patients, because they get a big immune response. So there are probably 3 different pathways to colorectal cancer and Lynch syndrome. One is you start off with a typical adenoma with a mutation in APC. Then as that adenoma gets bigger and gets more than 8 millimeters, it tends to lose one of whatever the relevant mismatch repair protein is, and you get a mismatch repair deficient adenoma, and you get a polyp that turns into a polypoid cancer. Down here in pathway number 3, you start off with one of these mismatch repair deficient crypts, and then you get a beta-catenin mutation, and you can get then stepwise carcinogenesis and get a flat cancer. But some of them start off this way, and then they pick up an APC mutation, and then you get a polypoid cancer. Now, what's interesting is this is the commonest pathway for MSH6 and PMS2. So they're polypoid precursors, and that's probably why colonoscopy is more effective in preventing cancer in these 2 diseases. Whereas in MSH2, this is the commonest pathway, where you start off with this defective crypt, and then you get a polypoid lesion. Whereas this pathway is not only common in MLH1, it's really rare, and it's less common in MSH2, but really rare in MSH6 and PMS2. So this is probably the reason why colonoscopy has a different impact, depending upon which gene is causing the disease. So if you take out the polyps all along, then you would expect to have a bigger impact with Lynch syndrome, MSH6 or PMS2, and less so with MLH1. and then in between with MSH2. Let's take a look at some variations on the theme. Muratori, constitutional mismatch repair deficiency, methylation-induced silencing of MLH1, Lynch-like syndrome, and familial colorectal cancer type X. So Muratori syndrome is simply Lynch syndrome plus specific cutaneous neoplasms. So sebaceous adenomas are fairly common, but they're more common in Lynch syndrome. Sebaceous carcinomas are very uncommon, and whenever you see a sebaceous carcinoma, you got to think about the possibility of Lynch syndrome. And there are also keratoacanthomas, an odd type of cutaneous tumor that you can see in Muratori syndrome. This is mainly seen with MSH2 and MLH1, and more common with certain pathological variants, rarely reported with the other two genes. Interestingly, this doesn't breed clean through families. So it's observed in 28% of Lynch syndrome families, and it's in 9% of all Lynch syndrome patients. So, you know, your Lynch syndrome patients might need to have a relationship with a dermatologist to look for these kinds of things. Interestingly, Muratori syndrome lesions have been reported with MUT-YH associated polyposis too. So there's a little overlap and a little bit of a confusing issue there too. And by the way, MUT-YH, as we'll see later, is related to a failure of one of the DNA repair mechanisms. A most unfortunate disease is constitutional mismatch repair deficiency. And that's when you get biallelic pathological variants. In other words, each parent has Lynch syndrome. And it's most common with PMS2 and MSH6. There's probably two reasons for that. These are the two most common Lynch syndrome genes. But I've heard it speculated, it's probably true, that maybe biallelic variants in MSH2 and MLH1 might be embryonic lethal. So in these kids, they get no expression of the mismatch repair gene. They get microsatellite instability in all their cells, and they get a childhood cancer diathesis with adenomas, cancers, brain tumors, small bowel adenomas, leukemias, lymphomas, really terrible. And you want to think about this if you're dealing with a population that has a high degree of consanguinity. And what's interesting is I've heard lectures in which people have gone to Iraq and Afghanistan, and in those countries, about 25% or more of the population marries a first cousin. It has to do with keeping money and property in the family. But when you have that kind of consanguinity, then you'll see more of these types of diseases. How about mimics and confounders? Okay, so the biggest confounder of Lynch syndrome is methylation of MLH1. So methylation of a gene promoter occurs at the CPG sites. That's cytosine and that's phosphodiester guanine. And about half of the promoters of our genes of our 20,000 genes have a CPG island. And typically this gets methylated, and it's a common way to permanently silence a gene. The progeny of the cells have this methylation, and it silences the gene. So you put methyl groups on the Cs at CPG sites and you silence it. But there are pathological examples of too little or too much methylation. So too little is hypomethylation, too much is hypermethylation. And the CPG island methylator phenotype occurs in a big proportion of colon cancer. So there's like excessive methylation, probably driven by BRAF mutations. And they have a different, the cancers come on later, but it's not a familial problem, but it's related to smoking and certain lifestyle issues too. So methylation-induced silencing of MLH1 at 15, if you look for microsatellite instability, 15% of colon cancers have microsatellite instability. But most of those, like 12% of all colon cancers, due to the acquired silencing of MLH1. You see this only in the tumor cells. We call this an epigenetic phenomenon, not familial. The tumors have microsatellite instability. They don't express either of these two proteins. And the patients tend to be older and more commonly women. And 90% are located in the proximal colon. Now that said, there's that's fairly common. This is rare. There's a rare issue of constitutional methylation of MLH1, where the methylation of one allele of MLH1 is seen in all tissues. So the first time I saw this, it was a 20-year-old woman. She had colon cancer. She didn't have MLH1 or PMS2 in her tumor. She didn't have a germline mutation, neither of her parents did. And when we looked at DNA from her hair follicles, from her cheek swabs, and her blood, everything had methylation of MLH1. And so this occurs in young patients, and it mimics Lynch syndrome. The tumors have microsatellite instability. They get all of the tumors that you might expect in Lynch syndrome. So you have to treat it like Lynch syndrome. It's usually sporadic with no family history, but Megan Hitchens, who's I think at Stanford now, or I think Stanford. Anyways, she reported that there was a sequence variant in the promoter of MLH1. And what that does is that triggers the methylation later. And interestingly, it runs through families because methylation is erased during germline formation, but this sequence variant promotes the, or leads to the methylation of the promoter. So this is really rare. Only a couple of families have been reported like this. A really much more common problem is Lynch-like syndrome. That's where you get two somatic mutations in the same DNA mismatch repair gene in the tumor, but there's none in the germline. Usually happens with MSH2 or MLH1. The tumors look just like Lynch syndrome tumors, but there's no germline mutation. This is probably about as common as Lynch syndrome. So there are a couple of explanations, and we'll get into this a little bit. It could be due to the fact that there's some germline mutation that leads to lots of mutations, and you get mutations in two of your mismatch repair genes. Or it could be a, quote, unfindable mutation, one that our current techniques can't find, the Lynch syndrome mutation, or a mosaic. The tumors are hypermutated. And interestingly, the age of onset of colon cancer is between that of Lynch syndrome and sporadic. So I think it's a mixture of some things we can't find and some that are genuinely sporadic. And the family history is somewhere between that of Lynch syndrome and sporadic colon cancer. It's probably as common as Lynch syndrome. The cause is uncertain. And I think we're in the next 5 years, we're going to learn a little bit about this. Historically, we would look at familial clusters and we said, okay, your risk for colon cancer is higher because you have a positive family history. And then we found the mismatch repair system. But what if you find a familial cluster and there's no germline mutation in a DNA mismatch repair gene, and the tumors don't show defective mismatch repair? Well, these families meet the Amsterdam criteria. 3 people in a family, one's the first degree relative of the other 2, and there's microsatellite instability in the, well, there's no microsatellite instability in this, but there's and it's not FAP. So no mismatch repair defect. And Lainey Linder from the Mayo Clinic coined this term familial colorectal cancer type X in 2005. The penetrance is lower and the tumors come on later. And multiple genes have been reported to create this phenotype. They're mostly individual family reports. And there only is occasionally a pathological variant found in more than one family, as we'll see in this next slide here. So every kind of familial colon cancer that's not polyposis could be called HNPCC, which was the term that Henry Lynch used to use for this. And in a really excellent review by Päivi Peltomäki, she noted that if you've got a group of people who have hereditary colon cancer, half have Lynch syndrome, half have this type X. The Lynch syndrome patients are younger, have a broader spectrum of tumor than colon cancer. The adenomatous polyp is the precursor in both. They're autosomal dominant. And here's our problem. There's too many genes here. The tumor characteristics are mismatch repair deficient in Lynch syndrome, but there's global hypomethylation in type X. So are there specific mutations that cause type X? Well, yeah. The problem is there's too many of them. These are just some of the genes that have been reported that cause familial colorectal cancer type X. So these are germline mutations, and you can see, so in this particular study, they looked at 272 patients, found this mutation in 7, 4 out of 118, and it goes on and on. And the problem is that these proteins and these genes are all over the place. And this is just if you look at the pathways, you've got DNA damage repair for some of them, but some of them are protein modifiers or developmental genes or RNA metabolism. You know, they're just all over. So we haven't gotten a handle on this. And the problem is that these genes that are involved here involve such a small proportion of people who have cancer that they're not yet on any kind of the gene testing panels. So this is going to be a problem. Otherwise, if we put all these genes on a panel, we'd be doing a 200 gene panel, and we'd be getting a lot of results that we couldn't possibly interpret. Okay, so let's get into what is a hypermutated tumor and what does that imply clinically. So when you have microsatellite instability, you've lost your DNA mismatch repair activity, you get a hypermutated tumor. Well, first of all, they're different biologically. They grow faster, they grow through different pathways, as we discussed earlier. And interestingly, they're less likely to metastasize earlier. So mismatch repair deficient tumors, they tend to downregulate HLA class 1, and they get mutations in beta-2 microglobulins. So these cancer cells tend not to create an immunological problem. That said, the tumors have a lot of tumor-infiltrating lymphocytes. But these defects permit immune evasion in the primary tumor mass. But it turns out if you don't express class 1 HLA and you get into the circulation, then natural killer cells will kill that cell. So I think that's why metastasis is more frequently local invasion, and people had a hard time studying how to treat metastatic Lynch syndrome tumors because only 4% of them presented with a stage 4 tumor. And that's because they would grow big in the primary tumor, they would evade immune detection. But once they got out into the blood, they'd be killed by natural killer cells. Didn't always work, but that was kind of the problem. The other thing too is that chemotherapy requires the mismatch repair system to kill the cell. In other words, the chemotherapy damages DNA, and then if the mismatch repair system can't repair all the damage, it just says, we're out of here, and it shuts the cell down and you get programmed cell death. And the most important thing, as I alluded to earlier, is colon cancers with microsatellite instability are immunogenic. They get these antigenic frame shift peptides. They get mutations that microsatellite sequences that give the same frame shift peptides downstream that attracts tumor-infiltrating lymphocytes. And that's why these tumors will respond to immune checkpoint therapies. So how many mutations are there per megabase of DNA in colon cancer? And are they different between hypermutated tumors and not? Well, the number of mutations in a colon cancer ranges typically between 1 and 8 mutations per megabase of DNA. And then there's like a little blip, and then it's between 10 and 100 for the hypermutated tumors, which are maybe 15% of all the cancers. Then if you sequence through all of the genes in the tumor, these are the target mutations in tumors, and it turns out that they're completely different between hypermutated tumors and not. This is a sporadic. This is 85%, and I really appreciated when I saw this paper. APC, P53, and KRAS are the most commonly mutated genes in sporadic colon cancers, which is exactly what Bert Vogelstein reported in 1989. And the only gene that's the same between these two groups here is the APC gene. So APC gene is mutated in 87% of non-hypermutated tumors, but only about half. So Lynch syndrome, half of them go through the APC and half of them don't. So they're really different kinds of tumors. So what are the clinical implications of a hypermutated tumor? This This study came out of Toronto in year 2000. It was a multi-institutional study, 607 colon cancers in people under the age of 50. And 17% of those had microsatellite instability, and presumably they were mostly Lynch syndrome. And the overall survival was 76% for the MSI high tumors versus 54% for those that weren't. And it wasn't because of it was a difference in stage. So 92% of stage 1 and 2 microsatellite unstable tumors survive for 5 years, as opposed to a smaller proportion in the sporadic ones that didn't have microsatellite instability. And even stage 3 tumors, there was a 70% 5-year survival. So they're really different. It's probably, I think it's because of this immune issue. So immune blockade was, what do they call that? Immune checkpoint therapy was initially tried on colon cancers. And I think the first 36 people they tested had no response except in one person. And I was at a meeting and this Dung Lee came up to me and she said, you know, that one person had microsatellite instability, and now we're looking at those patients and they're doing fairly well on immune checkpoint therapy. So this is the anti-PD-1 treatment. So tumor cells in Lynch syndrome are immunogenic, and there's a lot of tumor infiltrating lymphocytes coming into the tumor. So the tumor puts up a blocker, which is stops the immune response from killing the tumor cells. And if you give an antibody to that, then you can turn that around. And so when they in 2017, they reported that when they took these patients with microsatellite instability and gave them anti-PD-1, 21% had a complete response. And these are mostly people with stage 4 cancer, and they had a complete response. And it looks like those are stable responses. It's not like you just changed the curve. The people they're surviving for a long time. There were partial responses and another third. So more than half had some kind of an objective response. And it's all because these are highly immunogenic tumors. The tumors figure out a way to block the immune response, and this is a way to get around that. Now, the last thing I'm going to talk about are just the polyposis syndromes, because there's a few anecdotes that are kind of interesting about these. First of all, your pathologist is going to bail you out if they can recognize Port-Jaeger's polyps and juvenile polyps from adenomatous polyps. So the most common polyposis syndrome is FAP, caused by a germline mutation in the APC gene, but you can also get polyposis from mutations in these polymerase proofreading genes, polymerase D1 and polymerase epsilon. And then there are these recessives too, YH, NTHL1, MSH3. So you get 2 mutations, one on either side. The thing that you want to keep in mind about these recessive diseases is autosomal recessive diseases are completely genetic, but they don't look familial because of the recessive nature of the disease. And these all involve DNA repair genes. Then there are the hamartoma syndromes. Port-Jaeger's is caused by germline mutation STK11. Juvenile polyposis can be traced to 2 different genes. The P10 associated syndromes are more complicated. And then there's something called juvenile polyposis of infancy. And it's when you lose, it's usually by a deletion of these 2 genes, which are right next to each other on chromosome 10. C-rated polyposis syndrome, I spent a couple of years trying to chase this one down. It's usually not familial. We looked for everything, couldn't find it. And there are a couple of families that have germline mutations in this RNF43 gene. Now, there are 2 genetically based GI syndromes that were identified by their mutational signatures. One was Lynch syndrome, because people found microsatellite instability, but also MUT-YH-associated polyposis was linked to defective base excision repair. And I just think this is interesting because of what happened. So there were In Wales, there were some big families that had multiple siblings with multiple colonic adenomatous polyps, but there was no germline mutation in APC. So some poor postdoc had to sequence all of the APCs sequences in the tumors, and they all had 2 APC mutations, and they were all different. But there was a very common G to T or GC to TA mutation, and that suggested, and I won't get into the details here, but when the geneticists looked at that, they said this would be what would happen if you had a defect in the base excision repair system. So they looked for, they sequenced through the base excision repair genes and found the MUT-Y DNA glycolase had mutations. And so this was a disease that was found by virtue of its mutational signature. And that gave us a familial disease for all of the major DNA repair systems, like DNA mismatch repair gave us Lynch syndrome, and nucleotide excision repair gives us xeroderma pigmentosa, and base excision repair mutations give us YH-associated polyposis. And for what it's worth, BRCA is a defect in the homologous recombination genes, and that gives you the hereditary breast and ovarian cancer. Poit's-Jeghers and juvenile polyposis in Cowden's were found the old-fashioned way. So for example, Poit's-Jeghers syndrome was first characterized clinically, and then a genetic locus was mapped by the old-fashioned mapping techniques in 1997. And then finally, a serine threonine kinase was cloned and linked to Poit's-Jeghers syndrome, and most families linked to that. What I'm going to tell you in just a minute. In childhood, it's benign, but you get polyps that will obstruct or bleed in early life, but the kids don't get cancer. But Poit's-Jeghers syndrome is one of the most virulent cancer predisposing cancers. I think that 85% of Poit's-Jeghers syndrome patients will get cancer in their later life. Juvenile polyposis was confusing because pathologists were having a hard time defining what a juvenile polyp was, and it was initially linked to chromosome 10q. There was confusion about Cowden's and Banyan-Riley-Rubel-Comet syndrome. Our laboratory linked that disease to chromosome 10q back in 1997. But then 10q was suspected, but then ruled out for Poit's-Jeghers syndrome. And it turns out that juvenile polyposis, is caused by mutations in the TGF-beta superfamily, both SMAD4 and the bone morphogenic protein receptor 1A. That's where this comes from. So it's the same family of genes, but different genes can cause it. And then P10 and hamartoma, the hamartoma syndromes. Cowden's is interesting because it's the only disease that I know of that was named after the person who had the disease first, Rachel Cowden. That's probably some kind of a gigantic HIPAA problem, but that's the way the disease was named. And it was linked to P10 on 10q23, and it's a pretty pleiotropic disease, but also those same mutations can cause this childhood developmental problem. And the problem is that Cowden's polyps they look like hyperplastic polyps, but they can be other kinds of polyps too. One of the very last things I want to talk about is do these familial polyposis genes directly cause abnormal growth? No, they're really these landscaper effects. So Poit's-Jeghers syndrome, the kids get polyps which are non-neoplastic. They're characteristic polyps. They get large, obstruct, bleed, but they're not premalignant, and you don't need loss of heterozygosity. So the severe phenotype occurs later, probably due to loss of heterozygosity or that second hit. Interestingly, if you do a functional knockout of this gene in mice and target only the smooth muscle cells, they will get the Poit's-Jeghers polyps because the smooth muscle cells will induce abnormalities of growth of the epithelium. And you can also get the same phenotype if you target that gene loss in mice to the T cells or mesenchymal cells, and it goes through this IL-11 JAK-STAT pathway. And there's a landscaper effect as well in juvenile polyposis. So these Childhood juvenile polyps are non-neoplastic. The polyps have an inflammatory look to them. Cancers develop later in the polyp, unlike Poitiers. The cancers can develop in the stomach or the colorectum. But if you delete the SMAD4 gene in T cells in a mouse, you lead to upregulation of the inflammatory pathway, and they get these juvenile polyps too. So the polyps intrinsically are not neoplastic, but neoplasia may develop probably through the typical adenoma to carcinoma pathway. And so you want to keep in mind, it's a different issue. The formation of the polyp is different than the cancer occurring later in life. Okay, and the very last thing, this is the very last thing. What would happen if you did genetic tests on everybody with colon cancer? So Matt Yergelin at Dana-Farber took over 1,000 patients and did a 25 gene panel, a home brewed panel from Dana-Farber. They weren't selected for any genetic syndrome. And about 10% had one or more pathological germline mutations. 3% were Lynch syndrome. 7% had mutations in other genes. And this is kind of a problem because one of the reasons you test for Lynch syndrome is to know how to manage that patient, but the other is to how to manage the family. But families, some of them had APC mutations. This was a low penetrance variant, MUT-YH, BRCA, and these other genes too. And so what was problematic was neither the family history nor the age was helpful in predicting who was going to have these mutations. And this is what it looks like right here. No germline mutation, 90%, high penetrance mutations in 5%, and moderate penetrance in 4.7%. So this is the way we're handling this nowadays, is you do a panel test. But the problem is that you get Lynch syndrome in a bunch, but you're not sure about what to do with all these other germline mutations. And so I think that over the next several years, we're going to try to figure out what to do. What are you going to do about families with these kinds of moderate penetrance and lower penetrance mutations? And we don't know just yet. But this is what your genetic counselor is going to do for you. It's going to do a panel, and then you're going to come up occasionally with results that you can't really interpret. So that's what I've got to say. And if anybody wants a copy of these slides, I'll be happy to send them to you. Just email me at this address.
Great, Dr. Boland. Thank you so much. That was an outstanding presentation, and as promised, of Lynch syndrome and really beyond for all the hereditary syndromes that we take care of in the clinic. We now have some time for discussion, so I will review the chat box as questions come through, but please feel free to unmute your microphone and ask questions or make commentary based on Dr. Boland's presentation. I'd like to start with maybe a little bit of a personal question, a historical question perhaps. Dr. Boland, can you tell us a little bit about your family and personal connection to Lynch syndrome and why you got interested in this field to begin with?
Sure. So when I was a first-year medical student, by September or October, my father got sick and he had a colon cancer and he died of that less than a year later. And I started to dig into the family history, and it turned out that he had a colon, so he was only 49 when he died, but he had his first colon cancer at age 27, and his sister died at age 28 of colon cancer, and his father had a colon cancer at 25 and died at age 46, and his grandfather, my great-grandfather, I mean, everybody, as far as the eye could see, everybody had colon cancer. So, and they were young, and they tended to be in the proximal colon, and his sisters had endometrial cancers and colon cancer. I mean, he was one of 13, and if there's anything that a geneticist can learn from, it's from a big family. And so 13 kids, 10 of them had some kind of a cancer. So I went to my professors at my medical school and said, hey, this looks like a familial cancer. They said, no, there's really no such thing. The only familial colon cancer is familial polyposis. And I said, but what about this? And I gave them the story. And they said, well, colon cancer is a common disease. And so it's going to occasionally cluster in families. So I did my MD thesis on that. I did some laboratory research. This is 1971 and 72. You couldn't solve any genetic problem back then. And so ultimately I went into the laboratory with young Kim, and to study colon cancer, I was looking at glycoproteins, but that was like 1979 when I joined his lab. Well, 10 years later, genetic techniques allowed us to start to solve some diseases at a genetic level, and Bert Vogelstein kind of broke the ice and showed us how to do that. And so, well, then I got lucky, and I did a sabbatical, and I was studying microsatellites. I was using microsatellites to map for tumor suppressor genes, and so I had done probably 10,000 microsatellite analyses, and then bingo, microsatellite instability, which I had seen and didn't understand. I didn't know what it was. I took it to Francis Collins was there. It was at Michigan when I was there. We couldn't figure this out. Well, enough people got involved and figured it out.
You said you got lucky, but Carol, Dennis said that there's no such thing as luck. Do you remember his quote?
Luck has nothing to do with it.
I'm not so sure.
So there you go. I'm not so sure either, but that's what he said.
There's an outstanding question from Dr. Menard Ketcher, one of our luminal GI faculty, about early age onset cancer. I know this wasn't something that we talked about specifically, but can you talk about what we understand about the genetic spectrum in early age onset cancer, something that we're all hearing about and seeing in our clinical practices, and unfortunately even amongst our friends and family. Can you review what we know about sort of the genetic composition of early age onset cancer?
Yeah, and what's interesting is that a member of my family on my father's side and on my mother's side had early age onset colon cancer, and they did not have the mutation that was in my family. So, that one's personal too. Essentially, we started studying this 10 years ago, and the first question we asked was, is early age onset colon cancer all Lynch syndrome that we didn't suspect? And it turns out that there have been several studies, about 15 or 20% are Lynch syndrome, but 80 or 85% are not. So we did some additional studies looking for hypermethylation, like the CIMP issue. CIMP occurs in older people, not in younger people. So early onset colon cancer is not due to hypermethylation, but it is due in some proportion, but a significantly higher proportion. There's global hypomethylation that occurs. We don't know why that happens, but I'm guessing that the microbiome or diet or something like that is driving this. And when you get global hypomethylation, you get a more deadly tumor, in part because the hypomethylation allows the expression of these endogenous retroviral sequences, and also allows the expression of 3 different oncogenes. So you got additional drivers. So we don't know yet what it's all about, but biologically it's different. But it's not like it's a bimodal thing. I think that what's happening in the young people kind of blends into what happens with the older people. But hypomethylation is part of the story for some of them.
Right, and similar to the study performed by Matt Yergelen, Heather Hampel's group out of Ohio State did at least germline genetic testing and found that approximately 16% of patients diagnosed with cancer under the age of 50 have some germline genetic pathogenic variant.
Yes, and in that study, half of them were Lynch syndrome and half were something else.
Yeah. So Dr. Colgan just asked to maybe expand a little bit on some of the risk factors that you mentioned and what potentially the microbiome, the role of the microbiome in that second hit. Can you expand on what we understand about the microbiome and colorectal cancer pathogenesis?
Yeah, I think that probably one of the hottest areas right now in colon cancer research has to do with the microbiome. And certain bacteria, and a lot of this has just come out in the last year, certain microorganisms create a specific mutational signature. So certain E. coli and others. And so what's happened now is that people have taken colon cancers and done very extensive characterization in terms of mutational patterns, loss of heterozygosity, methylation, gene expression, all the rest of that. And there are like 80 some different mutational signatures. And most of them have not been linked to anything yet, but a bunch of specific microorganisms are being linked to specific mutational signatures. And also a study that came out in the last couple of months, they took tumors from various organs and then they sequenced they did the 16S sequencing. And it turns out that cancers of all, cancers of the brain, the breast, the pancreas, they have a resident microflora. They're sitting in there and they don't look infected and inflamed, but there are microorganisms, there are bacteria in them. And I think that we're going to eventually figure out how that resident microflora drives mutational accumulation. So I think that's a very important part of this.
Dr. Edmundowitz gets right to it. Certainly we have all experienced clinically that the barriers to genetic testing are becoming less and less. It's less and less costly. The access is increasing and improving. Why don't we just do comprehensive germline genetic testing on everyone the moment they're born?
Yeah, the moment they're born, wow. Well, it's interesting that when I started a high-risk colon cancer clinic in Baylor when I was there in 2003, it was about $1,500 for every test. So if you looked at MSH1 or MSH2, boom, $1,500. And then if you missed on that one, if you wanted to try another gene, it was another big amount. Nowadays, you can get for somewhere between $200 and $500, you can get enormous amount of testing, lots of different genes, a panel. In fact, some of the companies will actually, if you send your DNA in, they'll run like a 25 or 100 gene test, but whatever you ordered, they'll just report what you ordered because it doesn't cost them that much more to do the testing. So the reason for not testing everybody is we can't interpret, if we did entire, complete genome sequencing at birth, we'd probably create a lot of anxiety. Now, I might be wrong about this. I'm usually wrong when I try to guess these kinds of things, but we can't interpret so much of it. That's the problem.
And in our system, we give them a pre-existing condition, which might not be something that's protected long-term.
Maybe we can fix that in a couple of weeks, huh?
So, a lot
Dr. Patel, could I ask a question real quick?
Yeah, of course. Go ahead, Dr. Francis.
Thank you, first of all, Dr. Boland, for an excellent talk. I had a question for you about the immunology side of this with regard to those antigenic peptides that you were talking about. Do you think that those peptides could be used, if they are so antigenic, to develop like a vaccine against those specific types of cancers? Or do you think that the main benefit to that immunogenicity is in the Lynch or Lynch-like patients? Or would that be something that could be potentially beneficial to anybody to prevent colon cancer?
Yeah, definitely. Well, first of all, people with Lynch syndrome have been immunizing themselves their entire lives. So they already have the antibodies there. You just have to use the anti-PD-1 or whatever. You have to use to inhibit the shutdown of the immune system to get that to work. But that said, there's a fellow named Matthias Klor who has been trying to immunize people with these too. And I'm not sure what the point is because the colon's making these frame shift peptides all through a person's life. So I think that probably the major point is going to be to disinhibit the immune system in the patients with Lynch syndrome. However, for people who get an acquired microsatellite instability tumor, then that might be a very important way to maybe treat. So maybe there's a way to administer a vaccine together with immune blockade or something like that. I just heard a lecture today about that very that same concept. So we don't have a complete grip on this yet, but the fact is that tumors are screaming, I'm different. I've got these antigens that are not self. And I think the bigger point is going to be to figure out how to disinhibit the immune system rather than the vaccine. But vaccines are probably going to be more important for sporadic tumors, would be my guess.
One of the questions from the audience is about what we understand about the prevalence of some of these hereditary syndromes worldwide. A lot of the studies have been published out of Europe. Ong Lin's group is a little bit more international, but do you see populations, or do we have an understanding of populations where these syndromes have a lower or higher prevalence?
Well, I don't think it's published data on that. My guess is that probably the same everywhere, but there are going to be certain populations that won't get cancers as often. So Iceland is an interesting place because nobody moves into Iceland and Icelanders don't leave. And so they've got these huge founder effects. So it's like no MSH2 Lynch syndrome in Iceland, but they've got a lot of MSH6 and PMS2. So founder effects are probably really important when you have the lack of mixing of the population. So in the United States, we're all mixed, and so we're like the place to really to test this kind of problem. What's interesting is India has a remarkably lower incidence of colon cancer compared to almost anywhere else. And then Central Europe has a lot higher than anywhere else. And it's probably dietarily driven. So if you go to Japan or Korea, stomach cancer is a major part of Lynch syndrome. In Western Europe and the United States, it's so low that we don't screen. You screen maybe one time for stomach cancer, and that's it. And it's all driven by Helicobacter pylori. So even when you have these hereditary syndromes, there are lifestyle issues that probably drive the specific types of tumors that occur. And it's already been shown that if you're obese and you have Lynch syndrome, you're more likely to get a colon cancer. If you're a vegetarian and etc., then you're less likely to get colon cancer. So all of those things all play in one against the other. But I think that if you take a look around the world, probably the incidence of Lynch syndrome will be pretty constant, but different genes will be involved because of founder effects when a population doesn't outbreed very much.
There's been a lot of talk and a lot of interest lately in the pathogenesis of colon cancer and Lynch syndrome, and your discussion of these mismatch repair deficient crypts, and perhaps not the typical adenoma to carcinoma sequence, polypoid sequence that we have thought about for a long time. My question is, do you see, in addition to colonoscopy, in addition to chemoprevention, adjunctive screening techniques that may kind of harness that biology, either biomarkers in the stool, biomarkers in the blood that would have better sensitivity than colonoscopy at detecting these cancers?
You could theoretically look for microsatellite instability in DNA in stool. And Exact, the company that does the Cologuard test, tested that at one time, and then they got away from it because they didn't think there was a big enough market for it. Not the science is bad, it's just the economics were not there. And so theoretically, we could come up with a better fecal DNA test that could help you avoid too many colonoscopies. One time I was having a conversation with Henry Lynch, and he said, "Why not take the colons out of these people when they're 25? I said, "Oh, Henry, you can't do that." I said, "We have colonoscopy." He said, "Yeah. So you're going to do a colonoscopy every year from age 25 to 75. That's 50 colonoscopies." He said, "What is the downside to that?" And I said, "Well, you're right. There's a downside to doing 50 colonoscopies on somebody." Now, it turns out that that there's no difference in outcome between doing colonoscopies every year and every other year. I think it's a lot easier to get people to come back every year than every other. I think if you tell people to come back every year, maybe they'll come back every other year. If you tell them to come back every other year, maybe you never see them again. I don't know. So it would be really nice if we had a good fecal test, and maybe an FIT is good enough. I don't know. And that might be a good way to do it on the interval years.
Yeah, and certainly with the COVID pandemic, there's been some literature on using stool-based screening to triage.
Yeah. The nice thing about a good fecal test, like an FIT or whatever, is you could just postpone for a year or whatever that colonoscopy that you don't want to do during an epidemic. Now, in terms of the other things too, the gynecological problems are interesting because I got a phone call today from a physician a gastroenterologist in Dallas, and he said he had a patient who had Lynch syndrome, and her gynecologist took her uterus out in her 20s, which is not that's not a very attractive thing to think about. So typically we recommend having the uterus taken out once you've completed your family, having all the family that you want to have. I had a patient once who was also an MD PhD in Dallas who had Lynch syndrome, and we were talking about how to manage the gynecological problem, and the ovaries are one thing. And he said, well, if you're in the belly and you're going to be taking out part of the colon, he said, we said, why not take out one ovary? He said, maybe that would reduce your risk of ovarian cancer by half. And then, when menopause hits, then you could take the other one out through a laparoscopic approach. I mean, there's some creative space out there.
Maybe a little bit off topic from the content of your presentation, but we've certainly learned a lot over the last 10 years about chemo prevention in Lynch syndrome. Can you give our audience just an overview of what we know about aspirin in Lynch?
That's a really good point, and I didn't bring that up at all. Okay, so a guy named John Byrne in Newcastle, England, did a study where he took, I think, 800 or 900 genetically proven Lynch syndrome patients, and he gave them all 600 milligrams of aspirin a day. So that's 2 adult aspirin pills. I mean, European style. We use 325, they use 300. Gave them 600 milligrams a day. It was a fairly young population of people. And after 4 years and after 8 years, he sized it up, and there was no difference in cancer incidence. And he was about ready to give up. It was like, well, that didn't work. Actually, he was also using some kind of a resistant starch too. It was a 4-arm study. Then at 10 years, he was running out of money, and he took all the money that he had, and they did one final analysis. They gathered up all the data, and it turned out that the patients who were taking 600 milligrams of aspirin had a 40% reduction in all of the Lynch syndrome associated cancers, a 40% reduction, and a reduction in death too. But it took a long time. So subsequently, people have tried to reproduce this, and they'll report after 4 years that there's no benefit from aspirin. Well, yeah, it takes a long time. And what's even more important, and now they're studying to see whether a lower dose will give you some kind of a benefit too. 81 milligrams, 300 milligrams, who knows? I've been taking 300 milligrams of aspirin since the early 90s, by the way, when it was first shown that worked. What is really interesting is that we all assume that the reason aspirin prevents cancers in Lynch syndrome is because it inhibits the COX-2 gene. Okay, it does that, or gene expression. And actually, it inhibits the protein. But there have been a couple of papers recently that shows that aspirin modifies the microbiome too. I mean, this is really interesting. And it modifies it in a way, so there are certain bugs that are thought to be protective and certain that are thought to be causative of colon cancer. And the modifications in the microbiome go exactly in the direction that you would predict. So maybe that's part of it too.
What's really fascinating about the aspirin story is that just this summer, Sir John Byrne published the 10 to 20 year follow-up data and found that those same patients who were randomized to getting aspirin for only 2 years still had protective benefit 10 to 20 years out. And so, the kind of the timing and the mechanism is really, really interesting, and what really allows that sustained benefit.
Yeah, in fact, I kind of glossed over this, but in his first study that was in, you know, what's interesting is he was looking at familial polyposis and Lynch syndrome. And so initially he was looking at resistant starch and aspirin and an FAP, and it didn't work at all. And so that failed, that negative study ended up in the New England Journal of Medicine. Then he went forward and showed that aspirin prevented cancer in Lynch syndrome, and the New England Journal of Medicine wouldn't take it. So who knows? I mean, that just goes to show you how bizarre the publishing world can be. And so that ended up in the Lancet. But you're right. The people who took, I think he said it was a 4, as I recall, it was a 4-year, that the people who had at least 4 years out of the 10 when they were on aspirin, they had a significant reduction in cancer. So it just takes a long time, which is not surprising because colon cancer is typically a slowly progressing disease.
Thanks, Dr. Boland. We've had such an amazing discussion here. I do need to convey what I think is a compliment coming through our chat box. Dr. Kirkpatrick, one of our faculty at Denver Health, is just in awe that you were in medical school in the 1970s, and I was wondering, were you a teenager at that time?
No, and just to, if you want to make it even worse, my wife and I just had, as did Carol and Dennis, we just had our 50th wedding anniversary too. But I was pretty young when I got married, I have to say.
Fabulous. Well, Rick, thank you so much for the time you spent with us this morning, and I'm sure the fellows are going to enjoy interacting with you for the next half hour or so. Swati, thanks so much for organizing things, and to all the friends of Dennis, thank you for joining us this morning, and I hope you enjoyed Rick's presentation.
Carol and Nicole, this is for you.
Thank you so much. That
Thank
was
you,
really
Rick.
good.
We enjoyed it. So great to see you. Love to Pat.
Okay.
Congratulations on your 50th too. That's wonderful.